Okay, chapter 48 is the nervous system. So you need to know the brain structure. So on your review, I gave you a chart, so just make sure you know what those different parts of the brain do. I'm not going to go over them right now because you can write them down and just know what they do. You also need to know the different parts of the nervous system, so the divisions of the nervous system, I should say. And so we did that in class. I gave you that chart, so look at the chart, like with the central, sympathetic, autonomic, all of those. Look at chart we did in class. It's like a flow chart that you filled in the blanks and then we went over. Just make sure that you know that and know those different divisions and what they do, and so on. All right. Then, so that is just in general, the nervous system. And then you need to know, let's look at specifically the neurons. So understand just the flow of how we process information. So that is like the knee-jerk reflex. So look at figure 48.3 and 48.4. That is how we have sensory input, the integration, and then your motor output to your effector. So you have your sensory, and so on. And so within that, sensory neurons, what are interneurons? They're only in the brain and spinal cord, and then you have your motor neurons that go from the brain and spinal cord to stimulate a muscle. All right, so that's your motor neurons, and how these two are in your peripheral nervous system, and these are found only in your central nervous system, which is your brain and your spinal cord. So know the difference between that. And then look at that knee-jerk reflex, which was 48.4, and how that works. Okay, so review that. Also know the structure of the neuron. So you need to know all the vocabulary. So, dendrites, cell body, nucleus, axon, node of Ramvier, myelin sheath, a Schwann cell, a synapse or synaptic cleft, presynaptic cell, postsynaptic cell. So all of that, all those words and things like that, you need to know what they mean in a sentence. You need to be able to look at them and so on. So make sure you know all of that. So pause it and make sure, write all that down if you don't feel like you know it. So understand that, all right, and the supporting cells, or the Schwann cells, and so on. So study the neuron structure. Okay, so then you need to understand that neurons have what do we call resting potential. So, and remember that for a neuron, that resting potential is negative 70 millivolts, meaning that the inside of the cell is more negative than the outside of the cell. And you need to understand why is it at negative 70 millivolts. So what are the things that play a role in that? So we went over that in the notes, and we went over that in the nervous system tutorial. So thing number one is the sodium-potassium pump, right? Sodium-potassium pump. All right, if this is your dendrite, and this is your cell body, and this is your axon. All right, oops, sorry, I'm off the thing. Cell, dendrite, cell body, axon. All right, here's your sodium-potassium pump. Pumps 3 sodiums out for every 2 potassiums in, helping to make the inside of the cell be negative 70 millivolts. The second thing is the ion channels. Ion channels in the membrane. Which one is there more of, sodium or potassium? And you should say potassium. So therefore, these channels, remember there's more higher concentration of potassium inside, so there's more K plus going out than sodium channels with sodium going in. So you have more positives leaving than, because there's more channels, then you have positives entering, which contributes to that negative 70. And also remember that there are negative ions that are stuck in the cell. Why? Because there are no channels to allow them to be out. So then these negative ions are stuck in here. So all of that contributes to the negative 70 millivolts, which is the resting potential. So then you need to understand the whole action potential and what happens during an action potential. All right, so let's just go through that. I'm going to go ahead and turn this over and turn it sideways. This is what I did on the board, and we've talked about it for several days, but I'm going to go over it again. And so let's draw that big neuron. So you can hear it one more time. If you feel like you got it, you don't have to watch this. All right, and here's the synaptic terminal. Okay. So this is your cells. So remember that a stimulus comes the cell. If it's a sensory neuron, that stimulus is from the environment. If it is getting a signal from another cell, like a neurotransmitter, then that would be the stimulus. So here you have your membrane, and here is in the gated sodium channel here, and this neurotransmitter binds, the gate opens, sodium goes in, inside the cell becomes positive. So the inside of the cell becomes depolarized. So it was negative 70, now it's less negative, more positive, so like going to negative 60, then negative 50, and so on and so forth. So then that can trigger more sodium gates to open, it becomes even more positive. And so then how that works, remember, is you have your Schwann cells, your myelin sheath here, and so I'm just going to draw a couple here to show a couple of nodes. And so the sodium, this positive charge, once it gets positive enough, that's called the threshold level. When it gets positive enough, that triggers sodium gates over here to open. So the sodium gates open and sodium rushes in. So the inside the cell becomes positive. That triggers the sodium gates at the next node to open and the inside the cell to become positive. So that's what the purpose of this is, just to open the sodium gates at the next node. So then what happens here is next, I'll just draw it in maybe a different color here to illustrate it. It's a next. Happens afterwards. So that gate shuts. So the sodium gate shuts, and guess what opens? The potassium gates open, and K plus goes out. Why? Because it goes from high to low concentration. Remember, salty banana. All right, it's high potassium content inside, and so then the inside the cell becomes negative. That's repolarization. So this part became depolarized. Now it's becoming repolarized and going back to negative 70. Remember that potassium pump is a little bit slow to shut, and so therefore more K plus leaves than should. So it does get a little bit lower than negative 70, but remember your sodium potassium pump fixes that and pumps, and therefore you don't have a problem. Sodium potassium pump, very important there. All right, so the potassiums leave. Eventually this gate shuts and all is good. And so this goes back to normal. So now let's go over here. So this sodium over here, that positive depolarization triggers the sodium gate at the next node to open. Sodium gate at the next node to open. The inside the cell here becomes depolarized or positive. Oh, and then that it's done its job. It's depolarized. Now it's going to repolarize. Sodium gate shuts. Potassium gate opens, and K plus leaves from high to low concentration diffusion. Inside the cell goes back to negative again. Potassium gate shuts, and it's repolarized. Now let's look at the last node. The last node, we don't have to transmit that signal to the next node. You've got to trigger something to happen in the synaptic terminal. And so here you have vesicles in the synaptic terminal that have neurotransmitters in them. And so what happens is that positive depolarization at the end triggers not the sodium gates to open, but it triggers calcium gates to open. So there are calcium gated channels that are normally shut that open and allow calcium 2 plus into the cell, making the cell become further depolarized because of this positive charge. So what that triggers to happen is that synaptic vesicle fuses with the membrane, and out comes your neurotransmitters. So now your neurotransmitters can communicate with the next cell. And so let's say this is the dendrite of the next cell. This is the next cell. So this is cell 2. This would be the postsynaptic cell. This is the presynaptic cell, and this is the synaptic cleft. And it diffuses across the synaptic cleft where there's a ligand-gated sodium channel where the ligand or the neurotransmitter binds. That gate opens, sodium goes in, inside of the cell becomes depolarized, and the whole thing would happen again in the next cell. So that would trigger the nodes at the first to open and close, and so on. Now remember, this whole thing started out with the neurotransmitter over here. So the next thing you need to know is what happens to the neurotransmitter. It's usually either taken up back by the cell that released it. So that means like this neurotransmitter over here would come off and be taken in through endocytosis back into the cell, or there are enzymes that will break down the neurotransmitter. So you need to understand that. So how the neurotransmitter is kind of gotten rid of, because you don't want the neurotransmitters to stay on there forever, because then you'd have your neuron firing all the time, and we don't want that, especially if it's sending a pain signal. You don't want that. All right, and so that's in general how the neuron works. So use your practice sheets that I gave you. Remember the others I put on Google Classroom, the answers to those practice sheets, and use the back side for practice, and make sure that you can explain this. And so when we look at a neuron, they often say that the signal is transmitted from node to node. It kind of jumps. It's called saltatory conduction. It kind of jumps from node to node. So they'll say like the signal goes like this from node to node. And that's why, because the gates are opening and closing at each node. And then one other thing that you need to review about it is the graph that shows an action potential. So make sure that you review this that we talked about and know what's happening at each section, and so on. And so we did that also in the notes, and also in your nervous system tutorial as well. So be able to relate this to the picture that we just talked about, and know that this whole process, this whole thing happens at one node, and then it happens here, and then it happens here, and that kind of moves down the axon. And the last thing for the neurons is how they communicate with one another. So, or not how they communicate with one another, how just some basic overview of neurotransmitters and so on. So in the notes I talked about different types of neurotransmitters, so review those. So example, we talked about acetylcholine. Acetylcholine is a particular neurotransmitter that is between a sensory or motor neuron in a muscle, but there are also other different types of neurotransmitters. So, so review that. And that's basically the nervous system in a nutshell and what you need to know. So, so you know, practice with the diagrams and stuff. Know those different parts of the brain, and that's the nervous system.